Isolation and characterization of carbohydrates. Carbohydrates are generally considered as substances made up of carbon, hydrogen, and oxygen in which the proportion of hydrogen and oxygen is the same as that found in the molecule of water. Carbohydrates are large groups of organic compounds occurring in food and living tissues and including sugar, starch, and cellulose. Activity 1.1 Isolation of liver glycogen. Glycogen. Glycogen is stored mainly in liver, up to 6 to 8%, and muscles, 1%. However, due to the greater mass, muscles contain about 3 to 4 times more glycogen than the liver. Glycogen accumulated in liver serves as a reservoir that releases glucose into the blood when the blood glucose level falls below the normal one. The objective for this experiment is to isolate polysaccharide from an animal source and characterize carbohydrates according to the qualitative test results. For the materials, 25 grams fresh chicken liver without the heart, then 10% and 5% trichloroacetic acid, and 95% ethanol. Then sodium chloride, ice, preferably kept in an ice bucket, and then a warm water, sand, and cheesecloth. For the procedure, Step 1. Weigh 25 grams of liver and cut into small pieces on a petri dish. Step 2. Wash a small amount of sand thoroughly with soap and water. Rinse with water repeatedly until the soap disappears. Then rinse the sand with 5% TCA. Then, place the mortar and pestle in an ice bath until cold. Step 4. Grind the liver tissue together with the washed sand and 25 ml of 10% TCA at the same time in a cold mortar and pestle. Step 5. Strain the liver tissue after grinding through cheesecloth and collect the filtrate. Step 6. Rinse the mortar and pestle with 5% TCA and pour the washing through the cheesecloth. Collect the filtrate and discard the residue. Step 7. Transfer the filtrate into a 100 ml graduated cylinder and then record its volume. Step 8. Transfer the filtrate in a beaker. Step 9. Stir the filtrate slowly. Add 2 ml of 90% ethanol per 1 ml of the tissue liver extract. Step 10. Allow to stand for a few minutes until precipitates floculates. If no flocculation occurs, add a pinch of sodium chloride and place your container in warm water until a visible precipitate appears. Step 12. Note and record observation.
The isolation of plant starch.
Plant starch. Polysaccharides are synthesized by plants, animals, and humans to be stored for food, structural support, or metabolized for energy. In this experiment, starch is a polysaccharide subjected to different tests. The objectives for this experiment is to isolate polysaccharide, specifically starch, from a plant source and characterize carbohydrates according to the qualitative test results.
The materials we used in the isolation of plant starch are a whole potato, carrot, cassava, and unripe banana, and then cheesecloth. The procedures are as follows. Peel off the plant covering, cut and commute the sample, add at least 1 ml of water and grind well. Mix well, add 30 milliliters of water and mix well. Strain using the cheesecloth. Discard the residue and then collect the filtrate. Let the starch settle. Decant the solution slowly. Wash it once with water using the decantation process. Transfer on a watch glass and air dry. Note and record your observation. The isolation of glycerol. The sample used was chicken liver. The appearance of the precipitate was small flocculates. And in the isolation of plant starch, the samples used were sweet potato, unripe banana, and rice, and the end product was white starch.
Activity 1.3 General tests for carbohydrates. Materials. Isolated samples from experiment 1.1 and 1.2. Molesh reagent. Unthrown solution. 0.1 iodine solution. And concentrated sulfuric acid.
Activity 1.3.1 Molesh test. Procedure. Take 1 to 2 ml of 1% carbohydrate solution and add 2 to 3 drops of Molesh reagent and mix the contents. Incline the tube and carefully pour 1 to 2 ml of concentrated sulfuric acid down the side of tube so that the acid forms a layer beneath the aqueous solution. Note the color formed at the zone of the two liquids. Activity 1.3.2 Anthrones test. Procedure. Place 1 ml of 1% solution to the isolate. Add a 2 ml of anthrone reagent. Heat and note the change. Cool and record the result. Activity 1.3.3 Iodine test. Procedure. Place 1 ml of 1% solution to the isolate. Add a few drops of 0.01 molarity iodine solution. Heat and note the change. Cool and record the result. The tabulation of results of the general test for carbohydrates. We'll go first to Molesh test. The actual result we got was violet ring at the junction for both the glycogen and the starches, which matched the theoretical result, which is violet ring at the junction. For the anthrone, the actual result we got was green color for both glycogen and starches, which was different from the theoretical result, which is blue-green. And lastly is the result of the iodine test. The actual result we got for the glycogen was brownish, while the theoretical result was yellow-brown. The actual results we got for the starches were brownish. The theoretical result for the starches are blue-violet.
Specific reaction test for carbohydrates. Siliwanov's test. Materials needed: a Siliwanov's reagent, 0.1 molarity sugar solutions of each glucose, xylose, and fructose. Procedure. Step 1. In separate test tubes, add 1 ml of each of the sugar solutions to 5 ml of the Siliwanov's reagent. Step 2. Immerse in a boiling bath. Step 3. Observe the color changes in each tube during the first 15 minutes of boiling. Step 4. Record the color and the time of formation for each. Specific tests for carbohydrates. Tabulations of results. Glucose and Siliwanov's test produce TR yellow and AR yellow. Xylose and Siliwanov's test produce TR yellow and AR yellow, while fructose and Siliwanov's test produce cherry red and red. Let's proceed to fillings test. Materials needed: fillings reagent A and B, a 0.1 molarity sugar solutions of glucose, fructose, galactose, lactose, maltose, and sucrose. Procedures. Step 1. Mix 1 ml of the fillings A and 1 ml of the fillings B. Dilute with 4 ml of water. Step 2. Boil the solution for 1 minute. The solution that should be seen should be clear blue. Step 3. Place 8 drops of sugar solution in a separate test tubes. Step 4. Add 1 ml of the mixed filling solution to each of the tubes. Step 5. Boil the tubes in a water bath for 5 minutes and note the changes produced.
Activity number 1.4.2.4 The Barfoed's test. It is a chemical test used for detecting the presence of monosaccharides. The materials used for this experiment are Barfoed's reagent and 0.1 molarity sugar solutions, specifically glucose, fructose, galactose, lactose, maltose, and sucrose. This is the procedure in performing the experiment. Step 1. To 3 ml of Barfoed's reagent, add 10 drops of the sugar solution. Step 2. Place in a boiling water bath for 15 minutes. Step 3. Remove and allow to cool spontaneously. Lastly, Step 4. After 15 minutes, observe and record the results. Activity number 1.4.2.5. Benedict's test. It is used to detect the presence of reducing sugars. The materials used for this experiment are Benedict's reagent and 0.1 molarity sugar solutions, specifically glucose, fructose, galactose, lactose, maltose, and sucrose. This is the procedure in performing the experiment. Step 1. To 2 ml of the Benedict solution, add 5 drops of the sugar solution. Step 2. Place in a boiling water bath for 2 minutes. Step 3. Let it cool and record the results. These are the tabulation of results for the specific test for carbohydrates in activity number 1.4, which are the Fillings test, Benedict's test, and Barfoed's test.